trying to keep myself busy, and it's not really hard to do because we got the animals to feed, and that's why I'm late every morning. I've also been doing work around the house, okay? So last Saturday, so this is Rainer, our dog, who's half German Shepherd and half Siberian Husky, and he's getting to be friends with the cat. And he's actually completely frightened of the cat because the cat dominates. We have an alpha cat and a beta dog. Even though it's half Husky and half German Shepherd. And then, like I say, I have been working on the toilets around our house. I installed the toilet on the left last Saturday. My son, who lives in an apartment here in town and goes to MSU here, helped me on that one. And this is the second time I've pulled this toilet, and this was yesterday. And yesterday's work outside of that bathroom. So I've been keeping myself entertained and trying to get it back together before my wife comes home. But hold on, I will. This is pretty exciting. So let's see. My wife said she's been visiting my parents in St. Louis. She just took off on a long road trip. Friends of ours in Ada, Oklahoma, so she stayed there on the way to St. Louis, and I was driving back, okay, so a lot of miles, and I wasn't sure exactly when she's coming home. And it says, on the road, coming home to you. So that's great. That is great. That's in time for Valentine's Day. She's got 13 hours to drive, so I'm praying for her that she gets home to her guy safe and sound. And then I get this crap back together before she gets home, right? So I made the bed the first time this week, all right? And I'm also, the toilet seats are going back down, so I'm kind of re-gearing. I've got dishes to do, but it's a 13-hour drive, and so I... Okay, now look at this as the scariest picture, right? Doesn't this look like a wolf? So that's a silhouette of our dog. He is so hairy. While we're taking him for a walk. Okay. So I finally uploaded your outline. I had all sorts of issues finding it, finding the right version. I was missing a mouse. So you have the outline of chapter 8 now? Yeah? On whatever it is, Canvas. And so we're about right here. And so this is a sequence-based information on where genes begin and where that promoter region is. It's like a claw at the -10 and -35 that the sigma factor clings onto. And so this is kind of where it's at, what's really happening biologically. Here's the -10 and -30 sequence that are recognized in this really, really interesting protein DNA recognition and interaction. A lot of specificity because it is telling this big clunky RNA polymerase that this is the gene that you want to express, transcribe, and translate today. So really cool figure, getting fairly realistic. And I think I covered this yesterday, or I mean Wednesday, where 3 phases of transcribing DNA into RNA, and you have this on your outline. And I only give you the very first step of this because you have had this in previous courses. And so here is RNA polymerase, the holoenzyme scanning DNA for promoter sequence. Well, really, it's the sigma factor that is scanning. The RNA polymerase doesn't have the intellectual capacity to scan. It is just a polymerase. Just give me DNA and I will polymerize RNA. It's the sigma factor that can scan and look for and bind to the specific sequences. And when it does, then you have binding to the promoter sequence forms the closed complex. So that's not really important, but it's the conformational change of the RNA polymerase molecule that changes once it engages. And I'm not giving you any information about the antibiotics. This is good information. It's different locations where antibiotics interrupt this process. But we have a whole chapter on antibiotics, or a big part of a chapter later on in the course. And so then the RNA polymerase now does its thing, and that is unwinds DNA and transcribes RNA, incorporating new nucleotides and making RNA. And so you can see now the sigma factor has done its job. It has put the animal to work to polymerize that particular gene, and the sigma factor is going to go lock onto another RNA polymerase and orient it to its transcription location. So that's the only part of the transcription process that I'm covering. It's just that first step, because I'm trying to kind of focus you on the role of these sigma factors to lead the polymerase enzymes to their genes to transcribe. Okay, there are actually 6, and there's probably going to be more, there's probably going to be 8 next year, classes of RNA molecules. And this is in part all of the, quote, junk DNA that was thought to be junk 10 years ago actually is transcribed into RNA molecules that are not transcribed into protein, but that does not mean they are dysfunctional. So we've got 3 new classes of RNA of these molecules that used to be thought to be a cell's waste of time. But that's not how biology works. That's not how the creation works. There's an awful lot of focus and will to live, kind of like I was talking about last time, in terms of being efficient. If you're not efficient, you're going to lose out. Okay, so here's your 6 classes. You'll recognize most of these. The first 3, certainly mRNA, ribosomal RNA, transfer RNA. But here's it's kind of, for lack of a better word, it's small RNAs. There's also SI. There's all sorts of classes of these, which we really don't have time to go with. But these are new and regulatory RNA that regulates transcription or translation. So, the ability to control when and where you transcribe and translate those genes really gives you sophistication in terms of being able to respond to subtle changes in the environment. tmRNA is where it actually frees ribosomes that are stuck on damaged messenger RNA. And catalytic RNA is a really interesting class because here is a nucleic acid that behaves like a protein enzyme. And so it's actually, it carries out enzymatic reactions. And so really expanding our understanding of the role and importance of RNA. And like I say, there's going to be more classes, and there's an awful lot of information under these small RNAs that we're not covering. We're going to see them in action, I think, in one example later on in the course. Okay, so again, I'm not going to be covering much of this. You have had the genetic code since the 7th grade, possibly. But let's make sure we're on the same page. There is 3 nucleotides that code for, that are specific for, one amino acid. And there's 64 possible codons, which actually just, and we're talking about triplet codons, that specify 61 amino acids. But there are, among those, codons that tell the RNA polymerase this is the end of the gene. So they're called stop codons. And there's also start codons. You know that the code is degenerate or redundant. And so multiple triplet codons of the nucleotides code for the same amino acid. And what's interesting is, this genetic code is universal across from virus to elephant to oak to protozoa. with very, very few exceptions. So this unity of life, this single layer of order is used across life. And so, no details here, but this is what we mean by the triplet codon. So here are two different triplet codons that code for the same amino acid, tyrosine. And these are useful when you're doing protein sequencing or nucleic acid sequencing to figure out what the code means. These cool genetic code bases. So just see the code, 3 nucleotides specifying for 1 amino acid. But I'm not going to be asking you what is the triplet codon that codes for aspartate. Okay, and again, this is, I'm just trying to kind of look at these molecules a little differently than perhaps you've looked at them before. The transfer RNA molecule is really key in now translating nucleotide language into amino acid language in the process of translation. And we use that word specifically. Their shape, the 3-dimensional, 2-dimensional structure of transfer RNA are really important. And it has 2 functional regions. And basically the 2 regions is the anticodon. So it has 3 nucleotide bases that recognize the triplet codon at one end of the molecule, and at the other end of the molecule is the amino acid. So just to look at the transfer RNA molecule is to see translation going on. So on the left is this important secondary structure that transfer RNA molecules form themselves into based on base pair matching. And so you can see C is pairing with G, and A, adenine, is pairing with uracil. And so based on old-fashioned base pair matching, it forms itself into this cloverleaf shape. So that's what's shown on the left. But on the right is where you can see, here is your anticodon, here is the nucleotide language that is then translated into amino acid language. And so that's why we call it translation. And so just to look at a charged transfer RNA molecule, so when we say charged, it means it has an amino acid on it, is to see the process of translation. So I'm not going to cover any of the steps of translation, but look at tRNA and see it happening. Okay, so the other key player in translation is the ribosome. And so we're just going to look briefly at the structure. It's this really incredible hybrid between ribosomal RNA and numerous proteins. So, we learned there's 4 classes of biochemicals, but there's a lot of interesting biology in terms of hybrids between the classes. So this is the hybrid between a protein and nucleic acid. Represents, kind of gives the ribosome, those proteins give the ribosome this 3-dimensional, more complex structure. Okay, so there's 2 subunits, and in prokaryotes, they're a little bit smaller than in eukaryotes. And they are 30S and 50S, which S stands for Svedberg units, which is how they are sedimented in a centrifuge differentially. But then when they link together, and so we're talking about the 30S and the 50S linking together, you get 70S. So I'm sorry, here's 30S, here is 50S, and then you get them linked together and you get a 70S. So the point is, and I wish I had a diagram, when these 2 components of the ribosome link together, it forms a channel similar to the channel in the RNA polymerase molecule for the DNA. There is now a really nice channel that messenger RNA fits right through. And it ratchets itself down the messenger RNA right in between these subunits. So it actually comes in over the thumb and one of the fingers, and then through the palm of what looks kind of like a catcher's mitt. And that's where the messenger RNA is, and that's why you need two subunits to kind of clamp it down. And then you can see figure 819, which we don't have a slide of, for the secondary structure of ribosomal RNA. The form and function, you know, is just a great example of why we love biology. Okay, so again, this is, you know, interesting level of detail that I'm covering. This is the big picture. So here is now upstream of these genes that are transcribed is the promoter sequences that the sigma factor recognizes. And so then you've got a double strand, and you either have a sense strand or a non-sense strand. And basically, you know, it's the sense strand that has the information for the genes that are transcribed. So only one strand is transcribed at one time. And so now you have the DNA being transcribed into messenger RNA. Here is a stop codon that signals, oh, this is the beginning and end of one protein. And this is transcribed all at once by the polymerase molecule. And it is the messenger RNA that has these stop codons that stops here and just translates, the ribosomes hit those stop codons and just translate protein A and protein B. And the reason why I'm kind of reviewing this again for you is to remind you that in prokaryotes, there's often 3, 4, and 5 genes that are transcribed all at once in what we call an operon. And so this is an operon of 2 genes that are both transcribed at the same time. And so it's an example of why bacteria are a little bit more efficient in terms of getting their proteins expressed. Okay, this is one of my favorite parts of this chapter, and it is introducing you to a class of proteins that are very important in our cells as well as in bacterial cells, and they're called chaperone proteins. And so they lock on to typically damaged proteins, and they chaperone them to protein fix-it shops where you can renature proteins. And so this is actually one chaperone that has 2 components. And so we're looking at a chaperone protein called GRO-EL that forms a subunit with GRO-ES, and I want to kind of explain how these 2 subunits work together. They basically form together to form a barrel, a hollow barrel, in which damaged proteins are repaired. So that's why it has a hollow center. And so, you know, the faulty protein is chaperoned through these, it has an open hole and comes out repaired, you know, after getting twisted and punched back into shape. And so I just started interested in what is going on in this chamber that takes these bent, misshapen proteins that are denatured and gives them another life, renatures them into their proper 3-dimensional form. So mostly, if not exclusively, this is not a chemical change. So that amino acid sequence is the same. It has just become unraveled. And it's just another example of how important that 3-dimensional structure is, because if you lose that 3D shape, you lose function. So no chemistry is happening inside this chaperone barrel. It's just physics, just pushed back into the right shape. The sequence is the same before and after. There's no new amino acids. So it's not missing 2, 3, 4 amino acids. It's the same amino acid structure. It's just unraveled. And denatured is another term for a damaged protein. And when you repair it, you call it renatured. Okay, so this is actually a diagram that is in the very first page of this chapter 8. And it's a really nice diagram, and so I went and found it and made a slide out of it. And so here is your damaged protein, which is basically unraveled. It's almost in its primary structure, just a string of amino acids that's worthless. A string of amino acids can't do anything. It needs that globular shape. And so just a few steps in this interesting process. Damaged, unfolded, or denatured protein is captured by the GRO-EL subunit. And this is this kind of like this 2-ring shaped protein. And then the binding of ATP, which is not really shown or identified here, causes the top of the GRO-EL to elevate. And so just look at sort of this compressed form of GRO-EL, and now this expanded form of GRO-EL. That's what ATP binding does. It just elevates. It spreads that barrel out. And so that's the second step, is this elevation of the GRO-EL subunit. That then, this spread out conformation of the GRO-EL allows the GRO-ES subunit to dock, to attach onto the top of that surface. Okay, then lastly, what does that docking do? The attachment of the GRO-ES subunit forces the denatured protein, or if you want to say chaperones the denatured protein into this center chamber of the GRO-EL barrel. And so now you see the damaged protein, and if you look, maybe you could look at this with 3D glasses and really see the three-dimensional structure, which you can on that protein data bank, the PDB data bank, that I showed you the other day. But it's a pretty reasonable rendition here. You can see that this is an open chamber that now is where, once that cap shuts down, it gets I'm going to say, not mangled, it gets strong-armed, it gets pushed, it gets compressed back into shape. So I say smash back into shape. The denatured protein is now renatured, in other words, fixed, and it actually is then jettisoned out the bottom of this barrel. So really, mechanically and conformationally, very interesting action that proteins are taking on each other. So GRO-EL and GRO-ES, of course, are proteins, but their main function is to repair their cousins and brothers who have gotten beat up one way or another, been exposed to too much heat. These were originally called heat shock proteins because they were discovered that when cells were heat stressed, the expression of these chaperone proteins, these heat shock proteins, were upregulated and they were synthesized. So they are actually a little bit more heat resistant than the normal proteins, and heat, of course, is a pretty easy way to denature proteins. So there's a lot of different chaperone proteins, and there's a photograph of one of them, another one here. But here's another look, and so all I'm doing is covering the A figure, because DNA K is another example of a chaperone protein, and I'm not really going to cover that. But in A is this GRO-EL protein that has ATP attached to it, and now you have GRO-EL, and then the cap is in red, the GRO-ES cap. And then if you look down the bottom, if you look at this complex from the top, you can still see there's a little hole from on the very top of this protein. But it's inside this little hole that damaged protein is introduced into that chamber where it's thrashed back into shape. Okay, so really important class of proteins, these chaperone proteins. One other context, and I think I have a slide for it, is as proteins are synthesized, coming off of translation by the ribosome, realize that that strand of amino acids is just that. It's a strand. And it needs to conform into that 3D structure. Well, some proteins need some help in attaining that 3D structure, and that's another context for these chaperone proteins. So that's why you can see here's the ribosome, and you've got this nascent, just recently born strand of amino acids escorted by DNA K to the GRO-EL protein. So we've described this process in some detail. But I want to let you know that in some cases, some proteins are so trashed that they just cannot be helped. And so the cell has this kind of triage. So I say chaperone proteins are on the hunt for any out of line, which means denatured or damaged proteins. So cell performs triage where proteins are repaired by chaperone-based GRO-EL refolding pathways, which we've talked about, which is going on in the middle. Or if they're not repairable, then they are just plain destroyed by a protease enzyme, which is on the right. Okay, so so some proteins can't be repaired, and so when they cannot be repaired, then they find themselves just plain digested by these protease enzymes. Understand, when you see this word, this is a general word for a protein that digests, that cuts apart other proteins. And of course, if you can't repair that protein, it's worthless. Well, let's just digest it. We can use its 75 amino acids to make it again, or to make other proteins. So those subunits, those monomers, the amino acids, are perfectly functional, and they're reused. So it's kind of an interesting choice that is made by the cell, and discernment between you're a total wreck, oh no, you're repairable. So that's why the text calls it kind of a triage. Okay, so I think the last couple sections is we're talking about basically protein trafficking. So we really have already initiated a discussion of that. How does the cell move proteins around in the cell? And so whether a lot of proteins need to be excreted outside the cell, either outside that cytoplasmic membrane or actually outside both membranes outside the cell. So they need to be secreted. So proteins that are destined for that cell membrane or outside that membrane in the envelope require these special export systems. And they are tagged, so the cell can recognize proteins that need to be extruded outside of the cell because of these hydrophobic signal sequences, which are about 20 amino acids long. So when the cell recognizes this hydrophobic set of amino acids, it realizes, oh, it must need to go to a membrane. In other words, it must need to be extruded or secreted outside the cell. And so those hydrophobic amino acids at the amino terminus of the protein are recognized by another protein called the signal recognition particle. And that's shown in this diagram here. So this is figure 35 in your chapter 8. And kind of starting, let's see if I have, I'll bring that in just a second. So in this approximately 6-step process, trigger factor allows the ribosome to complete synthesizing the pre-secretion protein. Okay, so here is the protein that needs to be extruded outside the cell, and the SEC-B protein recognizes that hydrophobic region and says, oh, this protein needs to be extruded outside of the outside of the cell. And so it is this, what we call in here the signal recognition particle. And one of the things that's kind of interesting, it's actually keeping it in its primary structure, keeping it in a linear structure by wrapping around it. It's not letting it go into that three-dimensional structure because it needs to be sent outside the cell. It's a lot harder to excrete a big fat globular protein than just a strand of amino acids. And so that SEC-B protein brings the protein to another protein called SEC-A. These are all abbreviating for secretory. It's going to be secreted. And so now it's delivered to SEC-A and SEC-YEG. So see these two proteins that SEC-B has delivered the strand of amino acids to. And this is where the action starts. The protein is pushed through the SEC-Y channel to the periplasm. So you can see the SEC-YEG is a membrane-bound channel. And it's pretty interesting. SEC-A sits there and like pushes, like a hypodermic syringe, the protein about 20 or 50 amino acids at a time. 20. So SEC-A, which is shown here, it's in kind of the orange, you can see it's pushing the protein about 20 amino acids at a time. You know, so if it's an 80 amino acid protein, it needs to be pushed 4. It needs to be syringed 4 different times to get the protein outside the cell. And this is where you can see, oh, it's advantageous for this to be a linear protein and not a 3D globular protein, because it fits right through that channel. Okay, and then lastly, so it's a worthless amino acid sequence right now that's just primary structure. So there's a chaperone protein on the outside of the cell to fold it into its 3-dimensional structure. And that chaperone protein, well, it's not listed here. That signal sequence, that hydrophobic region is cleaved, and in the periplasm, chaperone proteins now give it the 3-dimensional shape. So that's a nice example of traffic control. The cell wants some of its proteins outside in the periplasm, or plain extruded outside the cell completely, and it has means to recognize those proteins with those hydrophobic amino acids, and then this 6-step process to escort it to the membrane and syringe it outside, and then refold it. And now you've got a functional protein in the periplasm or outside the cell. We are going to then look at one more step in terms of getting it completely outside the cell, outside that outer membrane. So when gram-negative bacteria need to export proteins completely out of the cell, so we've mentioned these, cellulase enzyme that's degrading the big cellulose molecule are exoenzymes that are excreted outside the cell. And there's actually numerous secretion systems that are they're called type 1, 2, 3, 4, 5, 6. And we're going to be looking at one of them, which is type 1 protein secretion. So, certain proteins are excreted directly from the cytoplasm outside both of the membranes in effectively one step. Okay, so the example is this toxin called hemolysin. Hemolysin. It lyses red blood cells. And so here is your hemolysin molecule, which is then ATP-dependent transportation through two different proteins. And really, I'm only stressing this one that spans the outer membrane and connects to the inner membrane, called the Toll-C outer membrane channel. So this is the kind of the workhorse of this system that effectively, in one step, pumps this toxin completely outside the cell and to wreak havoc. So in an infection, this toxin now is lysing our red blood cells, and we are trying to coagulate and stop and clot the infection, and it's kind of a war between our ability to clot this infection and prevent it from lysing our red blood cells to hemolysis, the lysis of red blood cells. Okay, I want to quit a little bit early, but I think I can, I'll just get through this bioinformatics so I can give you your exams back. So, this is totally not doing justice to this topic. There's at least 2 courses in the biology department and 1 or 2 courses on bioinformatics in the College of Ag that Mary O'Connell teaches. But John Ju in our department teaches bioinformatics. And so this is the informational and computational effort to order the tremendous amount of information that is in DNA and protein sequences. And it's really grown from that. So, in other words, we've been able to sequence the entire genomes of organisms, and since 1998 up until a few years ago, we actually got more than 200 species that have been completely published. The human genome has been completely published, and so it's a huge field. And so then the area of bioinformatics is taking that information and comparing it for many, many different reasons. You can make predictions about an organism's physiology based on what kind of genes they have. You can do interspecies relationships, genetic relationships, based on DNA sequence homology. And in the case of bacteria, it's really important because a lot of bacteria cannot be cultured in the laboratory. So we just extract their DNA, sequence the entire genome, and now we know an awful lot. We know every one of their genes, and so we know a lot of those organisms that we can't otherwise get to grow in the laboratory. We annotate gene sequences, and that basically means we identify that it is this gene, and it carries out this activity. So annotate is, here's this massive amount of sequence. It requires then people or computer programs to identify, oh, this is a hydrophobic gene. It must be a membrane-bound carrier. It must be a transport gene. And so that's what you call annotating. And in doing this, you are identifying open reading frames. And open reading frames simply mean they code for a protein. So here's this strand of DNA that might be 400 bases long, and you do not encounter a stop codon, so that's why you call it an open reading frame, that says, oh, in this particular frame, it is coding for a protein. And so that's what an ORF, an open reading frame, is a sequence of DNA that encodes an actual polypeptide. Okay, and I think I'll pick this up Monday morning to look at those different reading frames. And